Good day, everyone. Our presentation is entitled Molecular Approaches to Cancer Diagnosis and Treatment. Today, we will talk about how cancer can be understood at the level of genes, cells, and molecules. Instead of looking at cancer only as a tumor or abnormal mass, molecular biology helps us understand what is happening inside the cell and how this knowledge can improve diagnosis and treatment. To begin, cancer is one of the most serious diseases worldwide. It is complex because it does not happen from only one cause. It may involve inherited factors, lifestyle, environmental exposure, and changes in DNA. Traditional diagnosis such as imaging and biopsy remains important, but molecular approaches allow doctors to go deeper by identifying specific mutations and biomarkers that drive cancer. So the objectives of this study or this discussion will be the following. First, explain molecular or cellular basis of these approaches. Second, review latest advances and real-world uses. Third, analyze challenges, ethics, and future trends. And the last one, align concepts with K-12 education curriculum, particularly on senior high school biology. So first, let's define cancer. Cancer is characterized by the uncontrolled growth and spread of abnormal cells. Normally, cells divide only when needed, but cancer cells ignore normal control signals. They keep dividing, may invade nearby tissues, and can eventually spread to other parts of the body. So how does cancer cells are formed? So in this slide, it shows the cancer formation in a step-by-step process. It usually begins when normal cells experience genetic mutations. These mutations may affect the genes responsible for growth, repair, and cell death. As more mutations accumulate, the cell becomes more abnormal, grows faster, and may form a tumor, and finally, it will metastasize. So let's come now to the factors that affect cancer formation. First, we have heredity. Second, we have the lifestyle choices. Third, environmental exposure. Fourth, genetic mutation accumulation. And finally, tumor. So what is the significance of studying cancer and its molecular approaches for diagnostic treatment? So the significance of this topic is that advances in molecular biology have transformed cancer care. In the past, many treatments were applied broadly, even if patients had different tumor profiles. Nowadays, doctors can study the molecular feature of the tumor and choose treatments that are more specific. This improves the chance of successful treatment and may reduce unnecessary side effects. So what is the core of cellular and molecular mechanisms? Cancer begins when cells lose normal control over the growth and division. So instead of responding to the body's normal signals, cancer cells continue to divide, which may also avoid cell death, invade tissues, and develop the ability to spread. So in this slide, this explains that cancer cells show a generalized loss of growth control, which means that cancer is not caused alone by one defect. Several regulatory systems are affected, including the cell cycle, DNA repair, cell signaling, and immune recognition. These combined abnormalities make cancer cells different from the normal cells. So what are the root causes of cancers? First, we have the gene mutations. So one root cause of cancer is gene mutation. A mutation is a change in the DNA sequence. Since the DNA contains instructions for cell function, changes in DNA can affect how cells grow and behave. So not all mutations cause cancer, but mutations in genes that control growth, repair, and cell death can increase the cancer risk. Another factor is the oncogenes. So normally, some genes help the cells to grow and divide when the body needs new cells. But when these genes become mutated, they may become permanently active. They act like an accelerator stuck in an on position, constantly telling the cell to divide even when it should stop. Another factor or cause is the tumor suppressor genes. Tumor suppressor genes work like the brakes of the cell cycle. They slow the process of cell division, repair the damaged DNA, or cause the cell to die if the damage is too severe. When these genes are damaged, the cell loses an important safety mechanisms. As a result, abnormal cells may continue dividing instead of being stopped. Another cause is the DNA repair genes. So these genes are very important. Every time a cell divides, DNA must be copied. Sometimes small errors happen during this process. So DNA repair genes correct these errors, but if these genes are defective, mutations accumulated. Over time, this can make the cell unstable and increase the chance of cancer development. Another factor is disruptive cell processes. Cancer also involves disruptive cell processes. One of the most important is the uncontrolled cell cycle. So normally, cells pass through checkpoints before division. These checkpoints make sure that DNA is not damaged. Cancer cells often ignore these checkpoints, allowing the damaged cells to divide repeatedly. Another is the faulty cell signaling. Cells normally communicate through signaling pathways. These signals tell the cells when to grow, stop dividing, repair the damage, or undergo cell death. In cancer, these signals become distorted. Cancer cells may produce their own growth signals or ignore the stop signals. This allows them to survive and multiply even when the body tries to control them. So, what are the characteristics of cancer cells? So, there are two major characteristics. First, the loss of specialization. So, cancer cells may lose specialization. Normal cells have specific roles, but cancer cells often becomes less specialized and focus mainly on dividing. Second characteristic is cancer cells may become immortal. They can maintain telomeres, which allows them to divide far more times than the normal cells. Also, through the process of metastasis, they have the ability to spread. This is another dangerous feature of the cancer cells, which is they can break away from the original tumor and move through nearby tissues. They can enter the blood or lymphatic system, travel to another part of the body, and form new tumors. This is one of the reasons why early detection is very important. So, what is the important key scientific discoveries about cancer cells? So, we have the human genome sequencing, oncogenes discovery, immune checkpoints, targeted therapies, and nowadays we have this AI assisted treatment. So now I will be giving the discussion to Sir Cesar, who will continue what are the key concepts and historical developments or discoveries about cancer cells.
Thank you, Sir Jero, for your explanation for the previous parts. For my part, I will continue by discussing the key concepts in cancer biology, including genomic instability, metastasis, immune invasion, and the breakthrough technologies used in cancer diagnosis and treatment. Next, so, the key concept in cancer biology genomic instability means that cancer cells accumulate many DNA alterations over time. This is important because the more unstable the cancer genome becomes, the more aggressive and unpredictable the tumor may be. It may grow faster, develop resistance to treatment, or become harder to control. Next, oncogenes and tumor suppressors. So in this slide, a simple way to understand this is to imagine a car. Oncogenes are like the accelerator, while tumor suppressors genes are like the brakes. In cancer, the accelerator may be stuck and the brakes may fail. That combination causes uncontrolled cell growth. Next, metastasis is the spread of cancer from the original tumor to other parts of the body. Cancer cells first invade nearby tissues, then enter blood or lymph vessels. After traveling, they exit into new tissue and start growing there. This process makes cancer more difficult to treat. Next, the immune invasion of cancer. Our system normally detects and destroys abnormal cells. However, cancer cells can develop mechanisms to hide from immune attack. They may use checkpoint signals that tell immune cells not to attack them. This is called immune evasion, and it explains why some cancers can grow even when the immune system is active. Next, the major historical discoveries. In this slide shows important historical discoveries in cancer biology. Rudolf Virchow's cell theory helped establish that cancer cells come from abnormal body cells. This was a major idea because it showed that cancer begins when normal cells become abnormal rather than coming from something completely separate from the body. Next, radiation and oncogenes. The discovery of radiation by Marie and Pierre Curie became the foundation of radiation therapy. Radiation can damage the DNA of cancer cells and prevent them from dividing. Later, the discovery of oncogenes helped scientists understand that the specific genetic changes can drive cancer. This discovery helped shift cancer treatment towards molecular targets. Next, the immune checkpoints. So these immune checkpoints such as CTLA4 and PD1 act like brakes on the immune system. Normally, these checkpoints prevent the immune system from attacking healthy cells, but cancer cells use these checkpoints to protect themselves. Checkpoint inhibitor drugs help release these brakes so T cells can recognize and attack cancer cells. Next generation sequencing. So, NGS is one of the most important tools in molecular cancer diagnosis. It allows doctors to analyze the DNA of a tumor and identify mutations. Once the mutation is known, doctors can choose treatment or treatments that match the patient's specific cancer profile. Next, immunotherapy therapy and CAR T cell therapy. This in this slide discusses the immunotherapy and CAR T cell therapy. So in this approach, a patient's T cells are collected and genetically modified in the laboratory. These modified cells are then returned to the patient's body where they can recognize and attack cancer cells more effectively. This shows how molecular biology can strengthen the immune response. Next, uh, liquid biopsies. This liquid biopsy is a non-invasive blood test that detects circulating tumor DNA or cancer-related materials in the bloodstream. This is useful because it does not require repeated tissue biopsies. It can help detect cancer, monitor treatment response, and identify reoccurrence earlier. Next, AI and machine learning. So artificial intelligence and machine learning are now being integrated into cancer diagnosis. AI can analyze pathology slides, CT scans, MRI scans, and mammograms. It helps doctors identify patterns that may be difficult to see quickly, making diagnosis faster and more accurate. Next. So other molecular techniques include FISH, PCR, DNA microarray, nanostring, and next generation sequencing. So for example, PCR can amplify small amounts of DNA so mutations can be detected. FISH can identify specific genetic changes in chromosomes. These techniques help doctors and researchers understand cancer at a molecular level. Next. So, genetic information and cell function. So, this slides reminds us of the basic biological principle. Genetic information determines cell function. So when mutation changes genetic information, the cell may stop following normal controls. This is why identifying genetic changes is very important in cancer diagnosis. So I will now turn over the next part to Sir Vincent.
Thank you, Sir Cesar. So, in the next section, I will discuss current advances and applications of molecular approaches in cancer diagnosis and treatment, including liquid biopsy, NGS, targeted therapy, theranostics, and gene editing. Next slide. So, again, liquid biopsy has continued to develop as an important cancer tool. It can detect circulating tumor DNA, circulating tumor cells, and other cancer-related materials from blood samples. So this is helpful because it allows doctors to monitor the tumor without repeatedly performing invasive procedures. Now let's have the NGS, or the next generation sequencing. So it helps identify driver mutations, while targeted therapy attacks the specific molecules or pathways affected by those mutations. So compared with chemotherapy, targeted therapy is more specific. It tries to attack the cancer's molecular weakness while reducing harm to normal cells. Next, now let's have the theranostics and AI functional analysis. So for the theranostic, it combines diagnosis and treatment. It means that the same molecular target used to identify the cancer can also be used to deliver treatment. So AI functional analysis adds another layer by helping predict how tumors may respond to drugs. Together, these tools support personalized cancer care. Now, for the gene editing, especially the CRISPR-based technology, it offers a possible way to correct or modify cancer-causing genetic defects. This is promising, but it must be used carefully. If gene editing affects the wrong target, of course, it may create unwanted effects. So safety and ethical regulation are very important. Next, now in medicine and clinical practice, molecular approaches are used in diagnosis, treatment, and monitoring. So for diagnosis, they help identify tumor type and mutations. For treatment, they help match therapy to the patient's profile. For monitoring, they help track recurrence or treatment response. So in research, molecular approaches help scientists map cancer pathways and test new drug targets. So they also allow researchers to build patient-derived models. So these models are useful because they make cancer research more realistic and closer to what happens in actual patients. So molecular approaches are not limited to cancer medicine. They are also connected to biotechnology, agriculture, and environmental science. For example, biotechnology uses molecular tools to develop medicine and diagnostic fields, or kits, for example. Agriculture uses them to improve crops. And for environmental science, environmental science uses them to study organisms and pollutants. Now let's move to the strengths, limitation, and challenges. So the major strength of molecular approaches is, of course, precision. They can provide higher accuracy, support early detection, and guide personalized treatment. So this means patients may avoid ineffective treatments and receive therapies that are more appropriate for their tumor profile. So however, there are also limitations. Molecular testing, of course, can be expensive. It may not be available in raw resource areas. So the data can also be complex and difficult to interpret. Another issue is tumor genetic variation, meaning different parts of the same tumor may have different mutations. Some key challenges: standardizing tests across laboratories, managing tumor evolution, protecting genetic privacy, and reducing the long time from testing to treatment planning. So these challenges remind us that, once again, technology alone is not enough. We also need trained professionals. clear guidelines and ethical safeguards, and accessible healthcare systems. Now let's move forward to the traditional method versus the molecular diagnostics. So traditional diagnoses include imaging, tissue biopsy, and histopathology. So these methods show where the tumor is and what it looks like. So molecular diagnostic naman goes deeper. So it asks what mutation or biomarker is driving this cancer. So this allows more specific and again personalized treatment decisions. Now I will pass the presentation to Sir Stephen for the future directions and educational applications. Sir?
Thank you, Sir Vincent. For the final section, I will discuss the future directions, the ethical and social implications, applications in basic education, and our conclusion for this presentation. So, next, for the future directions and societal implications. The future of cancer is moving toward prediction, early detection, and also personalized treatment and continuous monitoring. Instead of waiting for cancer to become advanced, molecular approaches may help doctors detect risks earlier and adjust treatment as the tumor changes. Next slide. For multi-omic integration and AI diagnostics, multi-omic combines different types of biological information such as the genomics, the proteomics, and metabolomics of the patients. This gives a more complete picture of the tumor. When combined with artificial intelligence, this information can help doctors to understand cancer behavior and identify better treatment strategies. For machine learning prediction, the machine learning can help predict tumor aggressiveness, grade, and response to therapy before treatment begins. This is important because treatment decisions can become more accurate. Instead of relying only on trial and error, clinicians can use data driven predictions to guide patient care. We also have the multimodal data modeling. Multimodal data modeling combines the information from pathology slides, radiology, and molecular markers. By can help recommend customized treatment sequences. And in simple terms, it simply means it connects what the tumor looks like, it is located, and what changes it carries. Next slide, please. For the advanced liquid biopsies and longevity monitoring. Advanced liquid biopsies are expected to improve long-term cancer monitoring. By tracking circulating tumor DNA, doctors can monitor diseases progression without repeated invasive surgeries or tissue biopsies. This makes monitoring safer and more convenient for patients. We also have the minimal residual disease or the MRD. The minimal residual disease refers to the microscopic cancer cells that may remain after treatment. These cells may not appear on standard scans, but high sensitivity NGS may detect them. These can help doctors anticipate relapse earlier and adjust treatment before the cancer becomes visible again. So we also have adaptive sequencing. It is important because cancer is not static. Tumors can change, can adapt, and develop resistance mechanisms. This repeated sequencing allows doctors to monitor these changes and adjust therapy in real time. This makes cancer treatment more flexible and responsive. We also have the next generation therapeutics. It includes approaches such as intelligent reversion therapy. So instead of only killing the cancer cells, this approach aims to guide cancer cells back toward a non-malignant state through an epigenetic modification. This is still an emerging area though, but it shows how cancer therapy is becoming more advanced. We also have the in vivo gene editing, or wherein it uses the CRISPR platforms to target and correct cancer-driving mutations directly in in the genome. This technology has strong potential, especially for inherited cancer-related mutations. However, it must be carefully controlled because, of course, given that editing a DNA requires high precision. We also have the nanomedicine and smart delivery. It uses a very small particle to deliver drugs or antibodies directly to cancer cells. The goal is to improve treatment accuracy and reduce the side effects, and of course, help drugs reach the areas that are difficult to access. This is another example of how molecular science supports precision medicine. For our ethical, legal, and societal considerations, with the given of the power of the technology, ethical concerns become very important. Genetic privacy is, of course, a major issue because molecular data is deeply rooted to personal. Informed consent is also necessary. Patients must understand the purpose of molecular testing, its limitations, and how the results may affect their treatment and family. We also have challenges in equity of access and legal regulation. So, in advanced tests and treatments are often expensive, so not all patients, of course, may benefit equally. Legal regulation is also needed for genetic data ownership, gene patenting, and safety standards, especially for technologies like gene editing. It also includes the psychological impact. Molecular test results may also affect our patients emotionally. Some results may reveal inherited cancer risk or uncertain findings. This can cause, of course, anxiety and may influence the life decisions. That is why genetic counseling and compassionate communication are important parts of molecular medicine. So it includes, we also have in the next slides, the potential impact on science and the society. Molecular approaches can transform healthcare from reactive to proactive care. Advanced biological knowledge helps scientists understand gene function, cell behavior, and disease mechanisms. This impact goes beyond medicine because it also shapes research, policy, and public awareness. These technology influence public policy because governments need laws on health data, biotechnology ethics, and patient protection. They also promote scientific literacy, of course. When people understand genetics and biotechnology, they can make better health decisions and avoid misinformations. So how it applied to our basic education? These topics can be integrated into the senior high school curriculum, especially in the biology, under the special content on DNA, genes, mutations, cell division, genetics, biotechnology, and human health. Cancer is a meaningful example because it shows students that molecular biology is not just theoretical. It is directly affects diagnosis, treatment, and real-life healthcare decisions. These topics supports by topics in general biology too. Students can learn the structure and functions of DNA and genes, how mutations affect cells, and how gene expression relates to diseases. They can also discuss biotechnology in medicine and evaluate scientific evidence related to health and disease prevention. It may also incorporate it in laboratory exercises and simulations. So for classroom activities, students can do DNA extraction from strawberries or conduct gel electrophoresis simulations and PCR virtual laboratories. They can also use online simulations to observe the DNA mutations, cancer cell growth, and targeted drug action. These activities make better abstract molecular concepts easier to understand. To conclude, molecular approaches have transformed cancer diagnosis and treatment. They allow earlier detection, more accurate diagnosis, and personalized therapies that improve patient outcome while reducing side effects. Technologies such as NGS, liquid biopsy, targeted therapy, immunotherapy, gene editing, and AI, and also nanomedicine will continue to shape the cancer care. For our references, so we have the next slide. This final slide shows the references used in our presentation. These sources support our discussion on cancer mechanisms, molecular diagnosis, treatment and technologies, and future directions. Overall, cancer is not only a disease of uncontrolled growth, it is also a disease of mutation, disrupted signaling, immune evasion, and molecular changes. By understanding cancer at the molecular level, we can diagnose it earlier, treat it more precisely, and educate future learners about the value of science and healthcare.
So, that concludes our presentations on molecular approaches to cancer diagnosis and treatment. So through this topic, we learned that molecular biology plays a major role in understanding cancer, detecting it earlier, choosing personalized treatment, and monitoring disease progression. At the same time, these technologies must be used responsibly, ethically, and with attention to equal access. Thank you so much for listening.